Hello, everyone. Good evening. Can you hear me? So please put it down in the chat if you can hear me before we begin this session. I hope all of y'all are doing well. It's pretty warm this Tuesday evening. So please put it in the comments if you can hear me. Yes, good evening. Good evening, guys. Welcome, welcome. So good evening, everyone, and welcome to today's webinar on Bioinformatics for Molecular Docking and Drug Discovery, Top Computational Tools and Techniques. I'm Dilukshi, and I'll be your host for this session. So you've all followed us through the various webinars that we've had based on bioinformatics, AI/ML, data science as well. And we've looked at other aspects of bioinformatics, but today we'll be looking at the molecular docking and drug discovery side of things. Okay. So we are excited to have you here as we explore how computational tools are transforming drug discovery. So the hot topic for today is computer-aided drug design, or CADD, which plays a key role in identifying promising drug candidates faster and more efficiently. So it helps researchers in reducing the costs, and also it improves the precision. So today we have an insightful session planned with you all. So we have a range of expert speakers who will cover the fundamentals, the key tools, and advanced applications in molecular docking and drug discovery. So I'm sure you've heard of these terms like molecular docking, drug discovery, and all. So we'll be looking into more depth in this session. So before I talk about the speakers, so what is the importance of CADD in the pharma and biotech sector? So CADD is a game changer in drug discovery. It helps scientists predict how potential drugs will interact with their targets, allowing for faster screening of drug candidates before it goes into the costly lab experiment stage. So by using molecular docking, molecular dynamics, and AI-driven modeling, researchers can improve the accuracy, save time, and make data-driven decisions in drug development. So you can see how important this is. And this biotech industry is ever dynamic, it's ever changing. So you need to be in the know-how as to what new things are coming up, where else you can, look into career-wise. So, that's why we come up with these sessions. So today we're looking at this side of things. So let's begin. I'll introduce the speakers now. But before that, please share this link with all of your friends, your peers, because we'll try to have an interactive session at the end. So if you have any burning questions about, any career opportunities or in general, anything to do with this webinar, you can have those questions in the comments and we'll answer them as much as we can. So kindly share this with everyone. So who are the speakers for today? So we are honored to have three distinguished speakers with us today. So first off, we have Mr. Prodyot. So I'm sure you've all heard of him. You've seen him as well. Hello, sir. How are you?
Hello, everyone. Yeah, hello. Hi,
Yeah.
I'm good. Thanks for asking you.
Yeah. So a little bit of about Mr. Prodyot. So he's an expert in CADD, bioinformatics, and genomics, and he has research experience, and he has worked in institutes like IGIB, CLRI, IIT Madras, and he has his work published in reputed journals like IEEE and Frontiers in Pharmacology. Okay. So next up, our next speaker for the day is Dr. Nilofer. I'm sure this is another familiar name for all of y'all. Good evening, ma'am.
Good evening, all of you. Good evening, ma'am.
Yeah. So a little bit about Dr. Nilofer. So she specializes in big data analysis for cancer omics research. She brings expertise in drug design, molecular dynamics, AI-driven treatment strategies, and multi-omics technologies. And last but not least, our special guest speaker for today, Dr. Abhishek. So let's have a quick hello. Hello, sir. How?
Yeah, I'm good.
Thank you so much, sir, for making the time to come on this session and share your insights with all of our subscribers. Thank you so much, sir.
Thank you.
So a little bit about Dr. Abhishek. So he's a project scientist at CCMB Hyderabad. I'm sure you've all heard of that institute. And fun fact, we've posted some project vacancies from there today on Biotecnika. So do go check it out. So Dr. Abhishek has over 10 years of research in cancer, drug discovery, neurodegenerative diseases. He has authored over 10 peer-reviewed research papers in reputed journals and has received multiple awards for his scientific contributions. His current work focuses on small molecule therapeutics for Lewy body dementia. Now, before we begin the session, I'll just give you a quick run through as to how this will progress. So, we'll start off with Dr. Prodyot. So, he will talk to you about the overview of molecular docking and simulations in drug discovery, the fundamentals of it, how bioinformatics can aid in drug discovery, any challenges that it presents. And then followed by, we'll have Dr. Nilofer, who will talk about the key computational tools and techniques. She'll give you hands-on insights into top docking tools, best practices for choosing the right tools, etc. And finally, we'll end the session with Dr. Abhishek talking about the advanced applications and certain case studies and what's the future of AI/ML in drug designing, any limitations, trends, etc. So let's begin with our first speaker, Mr. Prodyot. So over to you, sir, and I'll meet you at the end.
Thank you for giving me this particular opportunity to speak. Okay, so I will share my screen and we'll start with the presentation. Okay, yeah, thank you. So yeah, so hopefully everyone can see my screen. Yes, can I have? Can anybody confirm? Yes, is it visible?
Yes, sir. Yes, sir, it is visible.
Okay, thank you. So hello all of you. So welcome to another webinar. So today we'll be dealing with this particular topic that I will be giving an overview of your molecular docking simulation and drug discovery. So we'll learn about what is a CADD, that is computer aided drug designing and discovery. So what are the terminologies if you are new to the field? So what you understand by molecular docking and simulation, what are the differences in both of them, what are the limitations of each? Okay, so we'll be talking about those. Okay, so without further ado, let's get started. Okay, so my name is Banerjee and I am a bioinformatics scientist in Biotecnika, Bangalore. Okay, so let's get started. So first things first. Okay, so we'll get an overview of molecular docking principles. So let's get a pointer by our side so that we can point out whatever I'm talking about. Okay, so first is so if you have studied about enzyme kinetics, you must be familiar with this particular term, that is your lock and key model. Okay, so something similar we are looking when we are trying to dock. Okay, so either it be your macromolecule in the form of proteins or receptors. Okay, so we are trying to come up with a ligand. Okay, so that will later be called as a drug molecule, and that we try to bind it to the receptor. So there are multiple ways how it can bind to the receptor. So these are the ways basically. Okay, so first is your lock and key model. Okay, so where you can see that the receptor and ligand have, like, the receptor is having a void that can be accurately filled by the ligand. Okay, so that is your actual lock and key model, how key fits into a lock. Okay, likewise. Okay, then you have your induced fit models. Okay, so where the ligand and the receptor are like not in synergy. Okay, but so it will initially bind to that, but ligand when first binding to the receptor, so the conformation is not right. Okay, so there is you can see there is a certain bit of gap in between. Okay, but the ligand basically will induce the receptor to fit in that particular cavity, and later on we can have a change in the shape that is happening in the receptor to make a stable conformation. Okay, so that is your induced fit. Okay, then further coming on to your conformational selection model. So when you have competitive like selection, so here you can see, so there are multiple receptor that are there to one ligand. So ligand will choose according to the, obviously based on the competition, it will see that this is the best conformation it can fit itself into. Okay, so that wise it will basically get the best conformation and bind to it. Okay, so then coming on to what are the types of docking that we encounter. So what is docking, we'll come to that. Okay, so this is just the how the docking is actually happening, to just so that people who are like learning it for the first time, they can relate to it. So that is why this particular slide is on first. Okay, so you have your receptor and ligand. So how the docking can be, so it can be a rigid docking where both your receptor and ligand are rigid in nature, so they are not changing their shapes. Okay, so here you can see the receptor and the ligand are they are not changing shapes. Okay, so after binding also they retain or sustain their particular shape. Okay, whereas in flexible docking you see that the ligand is flexible whereas the receptor is rigid. Okay, so that means the receptor will not be changing conformation or its shape, whereas the ligand will be changing its shape. Okay, then you have flexible docking. Okay, so flexible docking where both receptor and ligand are flexible. Okay, and they will try to be flexible. Why? Because they will try to fit each other. Okay, so basically they want to get into a perfect conformation of binding. Okay, so that is it. So you can get so the reference also I have provided in case you need to go to the depth, because everything cannot be covered in this 10-15 minutes talk. So that is why the references are also provided of the published journal, so you can go through that and in depth you can gain the knowledge of it. Okay, so now coming on to the terminology that has been discussed very much, that is your CADD. So what is this? Okay, and then coming on to your docking workflow and where does this particular docking falls into this particular CADD or the simulation that we are talking of. Okay, so we can tell that CADD is a bigger umbrella under which falls your docking, your virtual screening, as well as your simulations. Okay, so what are all these terminologies? We will discuss one by one. Okay, so first let's discuss about your computer aided drug designing. Okay, so that is CADD. Okay, so as the name suggests, computer aided drug designing. Okay, so we are trying to design a drug with the help of computers. Okay, so for some of the processes we might be requiring a good computational power, so in the form of your supercomputers or your HPCCs when it comes to your simulation processes. But prior to that, for your docking and your virtual screening part, that is not certainly required. Okay, and nowadays we also have AI driven CADD process that can automate the whole thing. Okay, so first things first, so basically what are the two tangents that you can draw from the CADD, or it can be done. So basically CADD is not a workflow that cannot be manipulated or basically customized. It can be customized basically, okay, based on what kind of disorder you are taking, be it your cancer you are dealing with, neurodegenerative disorders you are dealing with, or a certain kind of viral disorders you are dealing with. Okay, so certainly there will be certain bit of difference in the whole CADD process, but the backbone of it will remain just the same what we are discussing. Okay, so there can be two types of computer aided drug designing workflows. Okay, so first is your structure based drug designing and second is your ligand based drug designing, okay, famously called as your SBDD and LBDD. Okay, so in ligand based drug designing, when we go to ligand based drug designing, basically when we don't have the structure knowledge and we have the ligand, okay, so and based on the ligand knowledge we have to proceed. Okay, then we do the QSAR modeling kind of thing, then your pharmacophore modeling can be done, and then we move on to the virtual screening part. But if we have the structure of both, let's suppose your protein as well as your ligand, for both we are having, initially it will not be called as a ligand, let's suppose leads or hits we are having a structure of, so we can go for structure based drug designing, as the name suggests that we need the structure of both in this particular case. Okay, then we will be trying to find the binding side of it. Docking and docking and scoring will also happen. So this is also flexible. You can do the virtual screening first and then do the docking. Okay, so yeah, so what is virtual screening? Virtual screening basically means, as the name suggests, virtually we are screening something. Screening just means filtering. Okay, so what we are screening, we are trying to filter out the leads. So initially we'll be having a lead library. Okay, so that lead library will be prepared from all the hits that we can gather. Okay, so let's suppose we have 10,000, 20,000 leads initially. So now we have to filter that which fits into all the parameters that are there. Okay, so what are those parameters? Mostly these parameters are two. That is, first is your drug likeness or druggability, that a particular lead candidate can be called as a drug, yes or no. Okay, so that is your drug likeness. For that we have many rules that we will come on to. Okay, so that is your drug likeness. Okay, so druggability also it is called as. Okay, then you have ADMET analysis. Okay, so that is ADMET. that is also called as your pharmacokinetic and pharmacodynamic studies that do happen. Okay, so that means your absorption, distribution, metabolism, excretion, and toxicity. So it has to fall into that particular, like it has to, what do you say, qualify all those parameters, because at the end of the day, those things will later be called as a leads, and those things will be considered for drugs, and it has to be absorbed by the body well, metabolized by the body well. Okay, so and should be excreted from the body in the right time, so that it certainly does not produce any toxicity and side effect. Okay, so the dosage is also defined by that. Okay, so many things are there. Okay, so not diving deep into that. Okay, but basically virtual screening we are looking at. Okay, so once it passes the virtual screening, then we can have the docking, and then we have the pose scores of that. Okay, docking scores, also called as your affinity scores. Okay, then you have the compound selection, lead optimization, and then basically after this comes your molecular dynamics simulation, which are, you can say, the in silico people's mice, basically, because we are validating anything using your molecular dynamics simulations. Okay, and what is the difference between a docking and a simulation, that we will talk of in the next slides. Okay, so let's understand the workflow of the molecular docking real quick. Okay, so first we'll be having, so here we are talking about mostly the structure-based drug designing. Okay, in which we have the like structure of the protein, and if it is having certain heteroatoms, so we'll be cleaning that particular protein for the heteroatoms first. Then we'll be preparing the ligand library. Okay, so that is again then virtual screening. This that happens. Okay, and in the molecular docking process, we'll be having the protein structure, ligand structure, then we'll be finding the docking calculations. Okay, then different docking poses, then we can visualize the results. Okay, and how would we select that docking score? Based on the delta G, that is your gives free energy. So minimum energy, more stable configuration. Okay, so in the negative connotation, the affinity score should be higher. Okay, so that means -8 is better than -6 kilocal per mole. Okay, because it's an energy function. Okay, so based on that, we'll be calculating the docking score. So what is docking basically? So let's suppose we have a protein by our side. Okay, and it has multiple contours. Okay, so why do I say contours? Basically because we can have cavities as well as pockets in that. Okay, so and we will define a particular box to it, so that where the docking tool will confine, or basically find that our ligand can be bounded onto, like basically the periphery of it, or which particular pocket it can bind to, or which particular cavity it can bind to, based on the affinity scores. So that is why affinity scores basically gives us a rough idea of how the binding can be. Okay, so that is it. And based on that, once we finalize that, based on the RMSDs, so if you have the 0 RMSD, so that is the better, because less fluctuation is the in the RMSD, that is your root mean square deviation. That means better configuration it is at, and it is more stable configuration. Okay, so and in a particular cavity also, you can have multiple poses of the same ligand. Okay, so that will be judged based on the affinity score, as well as your bonds that are forming in that. Okay, so for that we'll be utilizing certain tools like Discovery Studio and likewise. Okay, so this is the paper that you can refer to if you want to dive deep into the molecular docking tangent of it. Okay, so yeah, so for the people joining new, so it might seem a bit heavy to you. So for that particular purpose, I have in between, I have also uh, included some slides to make you understand in layman language, or in very easy language. Okay, like obviously what we are trying to do basically is to get a lock and key model. So basically your ligand is the key over here, and it binds to a rigid protein that is your lock. Okay, so visualize it like that. Okay, so what are the limitations of a docking? So basically it is very simplified. Okay, but other ways of docking can fix this particular problem, like your induced fit models. Okay, so that will take what into account? Basically it will take all the changes also into account. Okay, so that solves the limitation of it. Okay, then you have different scoring functions. Okay, so there are multiple steps into docking when you are adding charges to it, because anyways it is happening in a 3D space. Okay, so you have to provide charges to it, add force field to it. Okay, so then likewise, then you will be setting up a box inside which. So what is this particular box about? You're defining the software that where the ligand should be where the software should be locating that particular ligand and finding the best possible fit. It can be a blind docking. Okay, so in case of novel drugs you do, and if you don't have the protein active site by your side, so you can go for blind docking. Okay, otherwise you can be site specific also, that you know that this is the particular area I can dock my ligand to. Okay, so that will do what? That will save the computational power basically. Okay, so we want our things to be computationally less expensive. Okay, so that is the main motive of it. Okay, and we want to do the things faster also. Okay, so yeah, same thing. So then coming on to your scoring functions, so there are multiple types of scoring function, but the main thing that we rely upon is the binding affinity, that is followed by that is done by your delta G, that is your Gibbs free energy. Okay, so how much spontaneous that particular thing is. Obviously we are not talking about the reaction over here, so basically we are speaking of the in the negative connotation how much it is. So better what is a stable energy wise, it is more stable. Okay, so then we have the types of scoring function that are there. So force field based, it can be amber. Okay, so amber is a tool also, it is a force field also. Okay, so then you have empirical like your chem score is there, and knowledge based you have drug score. Okay, so some of the steps involved in that is your protein prep that you do preparation, so then your ligand sampling, then scoring and ranking. So that is the simple overview of your how a protein ligand docking will happen. Okay, so then so you can take a screenshot of it, or basically this is a this I have basically included to give you a perspective of what are the basic tools used in CADD. Okay, and how bioinformatics is helping in that, because all of the tools that you see over here are from bioinformatics and are used in different levels of CADD. Okay, computer aided drug designing. Okay, so for literature search you have PubMed is there. Okay, so much more are there, but these are the important ones that are being enlisted over here. Okay, for chemical search or the ligand library preparation, you can have your PubChem, drug bank, zinc for your structures. Okay, IMPAP database is also there for searching for the what do you say, natural molecules. Okay, that are there. Then you have your protein structure databases like your UniProt, PDB, your SCOP, CATH. Okay, so then you have your structure modeling databases. If you want to do a homology modeling, you can go for your Swiss model. Then you want to do a ab initio, you can go for ITSR. Then you have your AlphaFold, that is AIML based, that has the benefit of all of the different modeling ways that you have. Okay, so you want to identify the mutation in that particular protein structure, you can go for alpha missense also. Okay, then in docking you have your AutoDock. From the AutoDock suite you have AutoDock 4, AutoDock Vina, Pyrex, that is automated, which utilizes your Python in the background. Then you have Glide, that is from Schrodinger. Okay, then you have ligand fit. Okay, so in admit screening, that falls under your virtual screening part. Okay, so two things can be done using this particular tool, not only admit but also your druggability analysis, that is your Swiss admit predictor or admit lab. Okay, so 3.0 is the latest version that has come in. Okay, then for cavity analysis, like if you want to dive deep into the what you say protein particularly, so you can go for your couch meta server, castp web server. Okay, then for interaction plots you can go for discovery studio, ligplot. Then your visualization tools. Okay, these are like PyMOL, caviar, rasmol, likewise. Okay, so then you have your molecular dynamics simulation. For that you have freeware tools like GROMACS. Then you have paid softwares like Desmond from Schrodinger again. Amber is also there. Okay, yeah. So yeah, again coming back to what we have started with, that is your drug discovery pipeline. So in general terms, we'll be having a target, then we have hits, then leads to drug. Okay, so that is our agenda basically. Okay, so how to do that? Okay, so target identification can be done initially. For that you have to go for your lead library preparation. That can be done using your tools like PubChem, Zinc, Drug Bank, Dr. Dukes, Impact, etc. Okay, Dr. Dukes and IMPACT are particularly for your natural compounds. Okay, whereas PubChem, Zinc, and Drug Bank, you can find natural as well as like synthetic compounds, as well as some of the published drugs also you can find. Because obviously we can also do repurposing of drugs. Okay, it is not that only we can go for novel lead compounds, but we can also repurpose a drug. The drug which is already available in the market, we can change its purpose to be used for certain different conditions. Okay, so then you have bioinformatic tools like for genomic what do you say variance analysis. You can use your UniProt, ClinVar, ClinGen, then your DBSNP. Okay, for identifying SNPs and all. Okay, then structural genomics. In that, if you have to like model a particular protein, you can utilize AlphaFold or Swiss model if the protein structure is not available in the PDB or UniProt, and likewise. Okay, for your structure based drug designing, you require those, so you can model those using this particular tool. Okay, if you want to identify the cascade of reaction that it is hindering or some part is malfunctioning, so you can go for your pathway analysis using KEGG and Reactome. Then you can also go for your protein interaction, okay, using string and cytoscape. Okay, so I'm moving fast because time is obviously limited. Okay, so basically virtual screening cascade, as we have talked about. Okay, so druggability and drug likeness, under which comes many rules that you see, like your Lipinski's rule is there, Ghosh rule is there. Okay, so then Aher rule is there, then Pfizer, GSK, Golden Triangle, Paynes. Okay, so if you want to dive deep into this, obviously you have to take up you have to study some paper about it. Okay, or you can join our internship that where we are basically explaining all the things. Okay, hands in hand. Okay, in our courses, as well as if you take up the internship, we will be explaining all of the things, and you will be doing hands-on on this particular thing as well. Okay, so then you have PKPD studies, basically your ADMET studies, that is your absorption, distribution, metabolism, excretion, and toxicity. Okay, for which you can utilize the tools like Sanjeevani web server, that is a supercomputer server from your IIT Delhi. Okay, then you have your Swiss ADME and ADMET lab that is there. Okay, so what is the advantage that CADD is providing? So we are talking so much about CADD. Basically, it is it is providing us the like a cost efficiency as well as time efficiency. Okay, so what used to take for R&D or for screening multiple molecules, it used to take 5 to 7 years. Now we can do it in a few months. Okay, and now with the acceleration of AI ML based tools, we can further what do you say smoothen that particular process within a month or two. Okay, so yeah, so that is it. Okay, so the success story is of your one of the case studies that like the coming speaker will also be dealing with, that is your HIV protease inhibitor. So that will be dealt in details in the upcoming talks. Okay, so yeah, so then we have the challenges in computational drug designing. Okay, so getting it fast, basically accuracy versus speed. Okay, so if we want to go high on accuracy, so that is your simulation, but it is slow in nature. Okay, so why is it slow? Because it is computationally very intensive. Okay, and exhausting. Okay, because it takes multiple things into account, conformational changes into account. So that is why it is slow, because it has so many calculation to do. Okay, whereas the docking can give you a quick overview of how the things might work. So that is why it is fast but less precise. Okay, so some example would be rigid docking, that is fast, but the flexible docking that takes many things into account, like the shape change, that is slow in nature. Okay, so then you have ligand flexibility based. Okay, so conformational sampling like increases computational cost. So that is what we have also seen in the case of your MD simulation. Okay, because that takes many things into account, all kind of conformational changes. Okay, so RMSD, RMSF, RG. Okay, so that is why it is computationally more costly. Okay, expensive. So then you have your solutions. So solutions can be your ensemble docking or machine learning guided sampling that can be done to fasten that particular process. And nowadays we are having those cascades already. Okay, that we have also discussed in our previous webinars where we dealt with machine learning based tools for your drug discovery. Okay, So last but not the least, I won't take much of your time. So basically is the overview of molecular dynamic simulation. So before getting on to the things, so these are the applications of it. You can read. Okay, so what are the applications? But I will simply define what is the difference between your docking and a simulation. The basic difference is that the dynamics, as the name suggests, molecular dynamics simulation. Okay, prior to this, we used to have Monte Carlo. Okay, so but this is replaced by your dynamic simulation because it takes all the dynamic changes or basically conformational changes that are happening in the protein during the course of simulation. Simulation is what? Simulation, basically you are trying to mimic something. Mimic what? Mimic our human body condition, because at the end of it, whatever drug you are proposing will be acting inside the human body, and human body is having multiple things going on, like your it retains a certain bit of like a certain temperature, it has a certain pressure. Okay, so then there are, what do you say, we have electrolytes, so obviously we have certain charges. Okay, so those things are to be simulated. Simulated basically means we have to mimic those things so that we can know in actual human body condition how the drug will what do you say act with the protein. So in docking we have the analogy of how the affinity, so that is static in nature, whereas this takes the dynamics into account, that like what are the changes that will be happening. Okay, so that is why the more the merrier, like more the merrier in the terms of how much time you can run the simulation for. Okay, so the larger time you can run the simulation for, that will give us a clear picture of how the drug or the protein will actually interact, how many hydrogen bond or what do you say covalent bond formed in between, broke in between, or the protein is getting destabilized in that particular process, getting out of the box, this that many things will be getting in details. Okay, so that is basically, this is the most detailed study in silico that you can get. Okay, so that in dynamic simulation and free energy perturbation, that is there. Okay, for that you can use freeware software like GROMACS that have their there you can find all of the processes in a synergy also, like how you select your, like how you create your topology file, then you add your like you have your force field selection, then adding charges to it. Okay, then you have your ensembles, NVT and BT ensembles, then going on for your molecular dynamic simulation runs. Okay, so those things. Okay, but it is actually computationally very costly, so for that mostly we'll be requiring a HPCC. Okay, that is your high performance computing cluster, or a supercomputer. Okay, so that will do the job. Okay, because unhindered power supply is required, as well as like it can run for months also. Okay, depending upon the size of the protein and what kind of simulation you are running. So there are multiple types of simulation also. Okay, so protein inverter, protein ligand. Okay, so bipolar, so multiple things are there. Okay, so yeah, if you want to dive deep into it, obviously you can consider joining our course for that. Okay, so then you have the, so these are the applications basically. Okay, so you can, so what it takes into account, so basically thermodynamic changes that are happening. Okay, so you can so it considers the fluctuation and dynamic changes, so that is the USP of your dynamic simulation over your docking. Okay, so then free energy perturbation can be done. Okay, then your molecular modeling of particular protein, that can be obviously analyzed. Modeling is not done using this. Modeling you will be doing using any of the softwares that we have talked about, either your Swiss model or your AlphaFold, but you can validate those results using this. Okay, calculate the NVT, NPT, all the ensembles that are there, or then analysis like SASA, radius of gyration, then your RMSD, RMSF, hydrogen bond, in different kind of interaction that are happening in the form of your dihedral potential calculation, like hydrogen bond, van der Waals, Coulomb's potential. Okay, then stability of a particular wild type protein as compared to the mutant protein. You can run a simultaneous simulation in two systems, and then you can compare how the mutant protein is behaving as compared to the wild type protein, checking for stability. Okay, then check for the stability of the dock protein ligand complex, conformational analysis, and then you can also do protein cavitome and pocketome analysis. Basically you want to dive deep into the pockets. Is there any, like, what do you say, induced changes that is happening, like if a ligand binds to pocket A and that brings about a change in the pocket 4, let's suppose. Okay, so that might happen. So for that, you can also go for pocketome and cavitome analysis using dynamic simulations. Okay, so yeah, so that is all from my side. Basically, so basically to recap, we have basically discussed about docking principles, okay, bioinformatic tools, MD overview. So some details about it will be dealt by Dr. Nilofer. She will be demonstrating some part of it. Okay, so last but not the least, I want to finish my basically presentation with this particular quote, that is, the best way to predict the future is to create it. Okay, so that is all from my side. Thank you everyone for joining today's webinar. Thank you. Yeah, over to the next speaker, that is Dr. Nilofer. Yeah, thank you.
So thank you very much, Prodyot sir, for giving very good insights regarding the theoretical overview regarding what exactly mean by computer-aided drug discovery and what are the different types, including structure-based and ligand-based. So that was really good insights where we get to know how the theoretical aspects can be covered and followed with the practical things definitely to move ahead. I hope that you all of you have even explored about the previous slide which contained the tools which is used for the computer drug discovery or design part. So I'm going to continue where I'm going to walk through all of you with respect to a quick one web tool which is available, which is called as a SwissDock, right? Because it's an overview where we are going to show you how exactly you can perform the molecular docking using the online application directly, right? And in that context, we are going to see how we can use the SwissDock, where we can perform the molecular docking with respect to the protein and ligand using the some example, and then followed with that we are going to discuss about how we can even get to know molecular dynamics simulations, right? With respect to the protein and ligand complex. Once we get the best ligand which is get docked against the protein, how we can even explore and get to know the dynamic study can be performed, where we will get to know the actual protein ligand complex in the water condition is nothing but the human body mimicking in that context by providing the water conditions and then running the protein ligand complex in the simulation period of time. So then followed with that, we are going to see about the how we can do the interpretations part, but because that is really important, guys, right? Because this is all the things we have discussed with the theoretical context. But to represent our result, okay, to showcase and get the interpretations part, we need certain kind of tools. And in that conditions, we have one molecular visualization tools, which is called as a PyMOL. Again, it's depend on the Python based programming stuff. So majorly the researchers used to use the molecular weaver or the molecular visualization tool, where we can show the particular protein ligand interactions with respect to the amino acids which are interacting with the ligands, and definitely the pockets we can showcase accordingly. So I'm going to share the screen and we are going to proceed accordingly. The first thing we are going to see is regarding the SwissDock. So yeah, so I hope that the screen is visible to all of you guys, right? So can I get some quick response? Screen is visible.
Yes, ma'am, it is.
Thank you, ma'am. So as you all can see, guys, like SwissDock is available on web, like online. You just have to just type as a SwissDock, and then followed with that, you will get this home page, right? The web app which contain the information about like SwissDock, which provide you molecular docking, and you all can see, guys, like we have used this example today because ultimately the backend, the plugin which is used in this web app is the AutoDock Vina. So ultimately, we are using Autodock Vina based on the web application. And the same pipeline we are going to carry out with respect to get to know how we can perform the molecular docking by using the specific target ligand, is nothing but the drug of your interest, and followed with the protein target which you are going to use for the study. So here they have given the two different way docking with the cavities. So if you know the specific cavities, guys, is nothing but the pocket region, you guys can proceed for doing the docking, or else you can even proceed with docking with Autodock Vina, because Autodock Vina contain a specific pipeline which provide you a pocket regions, and then where you can input the specific x, y, z coordinates to perform your docking accordingly. You guys can even explore the different tutorials which is even get provided over here, so that you all of you get a better insights regarding how the exact pipeline used to carry out. If you just scroll down, guys, they have given a very simple way how you can proceed the molecular docking by using very simple 3 to 4 steps accordingly. So please have a look, guys. Like, we are going to click on docking with AutoDock V now. So you have to just click on this particular feature. Once you just click on this particular AutoDock V now, then you guys can see now it's get highlighted, right? And you have to proceed with some examples which is get provided. So the best way to learn something new, right? So make sure, guys, like whenever you are using any new web applications or as any method, any practical context, so try to go with the known examples, right? So that you will understand what exactly the pipeline is get provided. If you just go with the known example, you all guys will get a better insights, right? How to proceed with the standard pipeline, and then you guys can go ahead with your research targets which you are looking to study. Okay, if you just directly go with your targets of the research which you are looking ahead, so might be there will be some errors will occur, right? Because some of the specific file formats will get not expected over here. So in that condition, it's really good to first go through the tutorials, and followed with that, please refer the examples. Okay, so the first step which we are going to do over here in this SwissDock is to submit a ligand. So you have to submit a ligand. You can paste in the form of the SPICE file format. I hope that you all of you know about what the SPICE stands for. So SPICE is one of the file format to call for the ligand, the drug. It stands for the Simplified Molecular Input Line Entry System. Okay, or else you can even upload the MOL2 file formats or as PDBQT file format, which is very standard for the molecular docking using AutoDock Vina application. You can even proceed to sketch your drug. So if anybody is good in chemistry, okay, so those guys or those researchers can use sketcher. Ultimately, if you just click on that, all of you can see that it's contain a Marvin JS, which is again one of the application integrated with this pipeline, where we can use the JavaScript, Marvin JavaScript of Chemexon, where we can draw the ligand accordingly. Can you see that? Okay, one benzene ring. So accordingly, you guys can see that it do read the smiles over here, right? So every particular structure which we are trying to include over here with respect to the benzene ring or as the functional groups atoms accordingly, it will start adding the smiles format accordingly. So you can use this particular format, or else you can simply upload the one of the example which is given over here. So simply click on this example so that we can proceed and get to know how the step can be carried out, right? So all of you guys can see that here is one smile format with respect to the example which I have selected with this drug. And if you just scroll down, you will get a very beautiful structure which get represented in with respect to the Marvin view application. And we can even get to know like how exactly the drug look like in the 2D format. So basically it's a two-dimensional representation of the drug which we are able to see on the screen, right? For the better understanding, because as a bioinformatics applications, whatever the tools or the softwares we are using, so all the things we are handling on the real time is nothing but we are able to see the data sets virtually. We are going to understand how exactly the steps can be carried out for the better insights of our research findings. I hope, guys, you are getting the steps which we have done in the first, that is submit a ligand. So once you just click off one of the example from here, so the next step which is going to ask you is to prepare your ligand. So simply, guys, you have to click on prepare ligand. So it will take some couple of seconds to just cross check that whatever the input ligand you have given, it's a correct format or not. If it is not, then it will give you an error. Okay, and based on the error, ultimately we have to like update our particular input drug, and you guys can see that here it's get tick mark. So ultimately it's showing you that the ligand which we have given as an input, it's correct. So we can proceed towards the second most important part. So in molecular docking, there is a two important things we have to proceed. First is a ligand of our interest, right? And second most important thing we need is the target. So target is ultimately our protein. So we do have the database available in bioinformatics platform, which called as a PDB, Protein Data Bank. So which contain a 4-letter identifier of every protein targets. So simply we can call that particular protein 4-letter code ID, or else we can even search accordingly. So that is the best way, guys, what we can do from here. We can directly call the PDB ID. Followed with that, it will even ask you like which specific chain you want to give to do the docking, right? So if your protein is a complex which contain a chain A, B, C, or even D, E, F, so in that context, if a more complex protein to do the docking, it will take some more couple of time to do the simulations and even the docking. So in that context, to make very accurate docking, you guys can even give a specific chain where the docking is to be done. So that is again a best feature which is get provided over here. You can even add the heteroatoms if any kind of study or the references if you have used where you want to pull out this target and you want to perform the docking, then definitely you can select the heteroatoms over here. If you don't know what exactly it does mean, you can even explore with respect to this question mark. Then you will get a more insights regarding the targets. Or else if you have directly a PDB files or the PDB QT file, then definitely that files we can even upload from here. So as we have given a simple search like a ligand in the form of the drug name, and followed with that it has taken this file format. And the second thing which we have done over here is to submit a target of PDB ID. Then simply the second thing we have to do is to click on prepare a target. So prepare a target is again it will check that the protein of the interest which you have given it's in the correct file format or not. Yes, it has been shown like it's a correct file format. So definitely the third most important step is to define your search space. So search space is nothing but where we get the protein. As you all can see, guys, what a best structure get represented on our screen, which contain the search box center. It's nothing but the cavity region. Can you see that, all of you? So whenever I'm just moving my cursor accordingly, it do contain a box. So this box is contain a size of 20, 20, 20. It's nothing but the specific three x, y, z coordinates is provided, and this box is get appear in this particular area, in as you all can see, right? So here also the entire protein is available which contain the alpha helix beta sheets, but this particular target is get selected in the cavity on the basis of the this particular numbers which is given. If you just change this numbers accordingly, as you all can see, guys, okay, just for the representation I will just show you. So you guys can see that it will start updating your box, okay, which is nothing but the cavity box, and that is really important. So we have to know what is our cavity box. Without cavity box, the docking will not proceed whenever you are using AutoDock Vina, or else if you are using SwissDock to perform your docking study. So make sure, guys, you are using the particular docking based on the active sites or else the cavity sites, so that the docking can be done in a very smooth manner with respect to the specific region which you are providing. So that is region is considered as ultimately a cavity region to proceed your docking. Okay, so I hope that this point is clear to all of you, and more insights you guys can even get to know if you just explore with this particular tab. Okay, so a better representation, you can see that what a fantastic image get represented on the screen. Virtually we can see that the protein is there, and it do contain a box which is nothing but a cavity box. So the cavity box will help us to define the x, y, z coordinates, and followed with that we can proceed the docking. So there it do contain a hydrogen bond interaction. So these things we can get to know once the docking has been done. Okay, you can even make a surface protein. Can you see that, all of you? So whenever we are doing certain stuffs, right, so it make ourselves to feel like, yeah, we are doing something which is a real research things, right? And that is exactly what bioinformatics is playing very significant role, right? So whatever we are doing, we are able to see directly on the screen, right? And we are able to understand how exactly the entire protein look like, what kind of simulations we can carry out, what exactly mean by the box region, the cavity. And once it is done, the docking has been done, you all of you even going to see, guys, like which are the amino acids which are interacting with our ligand. So that is the best part we are going to see ahead. So next thing we all can see is nothing but select the parameters. So like select the parameters are nothing but here the once the box has been defined, so you can select the sample exhaustiness. It's nothing but how much time it will take to do the simulations. Okay, it's nothing but the docking process. So the cavity which we have given, so it will select accordingly, and it will just proceed with respect to the random initialization condition, where the specific cavity regions are given and four different outputs will get represented accordingly with respect to the single ligand. Means the four different conformations it will get represented accordingly. So once you just click on check parameter, again it will cross check like whatever the cavity you have provided, followed with the exhaustiness, the sample exhaustiness. So it will just proceed with further docking process. So remember guys, like it's a web-based tool. So whenever we are providing the sample exhaustiness, don't make higher, because ultimately it will take a huge time. Okay, because sample exhaustiness whenever we are using with the web, so try to make sure that you just provide a simple like 1 2 3 4 accordingly, or else unnecessary it will take a huge time to do your similar docking process. Okay, so as you all can see that it is given you a rough idea, like as you have selected the simulation sample exhaustiness time. So it will take estimated time of 1.22. It's nothing but around 2 minutes it will take to give you the results on your screen, right? So that is how it will proceed. The fifth most important step it's given is like if you're providing a huge time it's taking and many of the jobs are in the queue, right? And your docking things is again it will take some time, so it is better to give your mail id and even the job title for the same, and then you can simply proceed for start docking. Once you just click on start docking, you guys can see that the page will get redirected and it will show you what kind of inputs you have given. Okay, the submission date followed with the ligands which we have inputted. Okay, the target of PDB ID, the box centers which we have provided with respect to the box size, which methods we have used to do the docking. It's AutoDock Vina, and sample exhaustiness is given with respect to their estimated job time. And then it has provided you that this page will get redirect automatically with respect to the results when it's get done with the calculation. So you guys can bookmark this, or else you can directly find the mail where you have provided your email id with respect to the results. So the results will even get appear on your mail id. So that is again a better advantage of providing the inputs of the drug and then getting your outputs with the mail with respect to the results and the calculations for the same. So for a quick way of discussion, because we are running with our time, so I have done this docking earlier. So and with the same ligand and the protein target, okay. And you guys can see that the query has been given with the ligand target, okay, the method of AutoDock Vina, okay, and the parameters has been set accordingly. And you all of you can see guys, like again a best one of the best thing which is get provided is the research papers, the citations, right? Where we can understand exactly how this SwissDock is about and what are the different features is get added and how, what kind of writing part we can refer with this research papers. Okay, so this is our ligand which we have used, and if you just again scroll down, you guys can see that the results get appear, right? So this results is there which contain, to understand the 3D panels, they have given this 3D panel, the view, where we can display our structures using the AutoDock Vina application, which we have used ultimately. The one of the again best part which we can get from is that we can export the results in the zip file as well as we can attach towards the mail. Okay, and now to do the quick interpretations of the results, you can see guys, this is exactly what you will get the model. So these models are nothing but the single ligand is giving you a multiple conformation. And the next most important thing to make your docking results as a best is nothing but the calculated affinity, the binding affinity of ligand in the form of kilocalorie per mole. So the best ligand which is done docking in the first conformation is minus 9.79. So what you have to do simply guys, you have to click on this ligand. For example, you all can see that if I just click on this ligand, the first conformation, the ligand will get appear in the specific protein. Can you see that? Okay, so this is exactly where the hydrogen bond interactions are there, right? And followed with that, you can see the ligand is get buried, the ligand is get docked in the specific cavity region, the box region which we have provided in the chain A. That exactly how it will give you the results. You can see that this is hydrogen bond interaction, right? So if you just roll your cursor accordingly, you can see that the specific ligands which are interacting will get appear over here. And that is what exactly which we need in the form of docking study. Okay, valine at 441 position. Okay, 471 of chain A it's given. Here you can see that again isoleucine at 463 positions. Okay, from where I'm reading you can see here, right, it's getting appear over here, the atom and the amino acid and its position with the chain with respect to the atoms which are interacting. If you again scroll down, you can see that the hydrogen bond interaction, you can see that if I'm just removing this, so that is getting this selected. So basically it's a hydrogen bond interaction. Ionic interactions you can even get to know from here. So whatever the 11 yellow dotted lines are there over here, so those are the ionic interaction, whereas the blue dotted line over here is the hydrogen bond interaction, and that is really important, right? Then the cation pi interactions are given. So you can even get to know which are the pi interactions. Hydrophobic contacts, stacking interactions are even included for the docking study. And then definitely show protein surface. If you just click on that, you guys can see a best structure where the ligand is get docked in the cavity region. Can you see that? What a pretty picture which get appear on the screen. So ultimately this kind of images we can use directly for our paper publication, okay, or else any conference if you want to present your work, right? So you can guys can directly take the images where you can show the exact pocket region where the ligand is get docked, right? And followed with that you can show the amino acids which are interacting. So this is this different way of like doing your docking study, followed with that showing the cavity regions and then getting the specific interactions with respect to the docking scopes. All of you are clear with this? Okay, are you guys understanding? Right, definitely. So what do you say, guys? This is really fantastic things. Whatever we are doing with our input, we are able to get the output directly on the screen. Okay, and followed with that we can do a best interpretations for the same, right? With respect to the docking scores, the statistical analysis, you can see that whenever you are selecting any second ligand, so the second ligand is get selected, right? So whenever we have to take is that the first one. Okay, this is also a 2D way of representations you can showcase. And then for example if I just go with a very less or less docking score, right, for example this one, you guys can see that the pocket region, okay, which is it's very far, right, it's very outer towards the pocket, whereas the first cavity you can see that it's very inside, right, it is get buried inside with respect to the protein target. And that is exactly what we have to take the first confirmation of the model with respect to its docking score, and in minus it should be higher whenever we are doing molecular docking with respect to majorly with AutoDock Vina. So it is minus 9.97 791. It's nothing but the best docking binding affinity, and the first confirmer is the best confirmer which we use to consider. Okay, if you want to get to know more in slides, definitely you guys can explore the results. This question marks will ultimately give you the help panel to understand how to use the control panels of all the features which is get provided. Okay, so I hope that you all of you guys understood how exactly the docking step has been carried out, right? As for the next agenda, I'm going to show you the next application is the PyMOL, right? So PyMOL is considered as the molecular visualization tool. So whenever we used to get a representation, right? So in this context, we have to download the PyMOL. I hope the PyMOL is visible to all of you. The and what you have to do is nothing but you have to go in the file, and then there is a different options like you can upload or open your pdb or the complex what you have after docking, or else you can even get the pdb id directly from the pdb database. So I'm going to share like one quick target 4mq4. Okay, this is one protein pdb id of kinase target, and you have to like just download that thing. Okay, you guys can see that what a pretty picture get appear on the screen. Okay, and this is exactly what the publication quality image we need, okay, for our research papers or as a publication quality work. Okay, and here you can see, guys, like it is exactly what you can see, which contain the ligand, and this is exactly what the protein with respect to different phosphate group accordingly. Okay, how exactly we can do this interpretation and more insights for the same. You guys can see here it's get represented like A is to action, so you can just click on action present ligand site, and just you just mention like a cartoon. Okay, so all of you can see this one. The same thing is getting represented, but using the molecular weavers, using some more specific features. If you just click on this particular options, it will help you to give you a different representation and even the orientation of the ligand and their interaction. The second thing what you can do is that you just click on the particular options over here, where you can get the, so it's A is call for action, okay, S will give you the show hide, okay, and it will help you to label your specific targets accordingly. So the action it's done about like it has shown you the particular target where you got the particular present ligand, okay, and the ligand side we have click on the cartoon representation, right? So you guys can even make, can you see that publication quality targets we can include. Okay, we can even get to know like present uh, the ligands which are the interacting. You can see now only the ligands will get appear, and if you just click on the dotted line, right, so this is ultimately where the specific present, right, the ligand names you can represent. Okay, you can do the undo just by representing in this way. Okay, again the action you can do from here, and present ligand side, you just mention dotted surface if you just need mesh surface, whatever. So it will just give you a very nice representation of your ligand within the pocket region. So ultimately, this is a pocket region where the ligands has been included. Okay. Some more examples I have given over here like this. Um, you guys can even get to know the, yeah, the specific amino acids which are interacting. So many things we can do with the molecular weaver application, where we can view the ligand with respect to the pocket regions. We can showcase the amino acids which are interacting, and then we can even use this representation for the output directly. Okay, so this is the second agenda as for the today's quick webinar, and the last agenda which is get lined up is nothing but to show you the MD simulation part. Okay, basically MD simulation is nothing but it's take a huge time to do the entire simulation process. But if you just type GROMACS simulation, so you guys can find this quick link where the tutorials is get provided. But the best way is to explore the tutorials with respect to this page, which will give you a tutorial in lysosome in water, and it is one of the simple example to get to know how the particular target will run the simulations within the water conditions. Okay, with respect to your nanoseconds, because in body, as Prodyot sir has mentioned, like we have to take a model of the human body, certain kind of mimicking things we used to do, right? And in that conditions we are using this water, because in our body ultimately contain more percentage of water, right? More than 70% of water is there. So if we are taking any drug, right, because all the things we are doing to get to know like how will the drug will get fit into that specific protein, where we are targeting maybe for the activations or else to inhibitions of the specific uh, target what we are looking for. So in that condition the ligand should go and bind in that specific pocket region. So we used to study the entire process by using the molecular dynamics simulations as a step. So in that we have the example lysosome in the water, but that is a command line steps are there. So you guys have to explore step by step. So ultimately you will get the simple example lysosome in water. You guys can even proceed with protein ligand complex, what we have done right now. The same steps which is given with respect to how you can do the MD simulation using the GROMACS application. Okay, so the condition is that you have to install this thing. You have to go with respect to the command line, and more basic tutorials is even provided. but how it's looked like. I just want to show you guys like how exactly these things get shows with respect to like protein and ligand. Okay, you can see that it is exactly in the water condition. So basically it's a complex, right? And it is going the simulation in a very nanosecond, and this used to take with respect to the simulation times which we are providing. So that is called as a dynamics. Okay, basically the molecular dynamics means what? Molecular are in the dynamics condition. And then you guys can see that here whatever the box is given, so that is nothing but a box which contain a water. So the complexes get deep into the water condition, and there the ligand should not lose its binding, what you can say as a interaction. Okay, it should be stable. The protein and ligand should be stable. So that is exactly what we used to check in the protein ligand molecular dynamics simulation, using the GROMACS or else using Schrodinger Mastro. So that is exactly what we used to check, like how the protein and ligand complex is having a stability in the water condition. It should not lose its affinity, right? Because ultimately the drug should not lose its affinity, right? It should have to perform the specific function. For that purpose only we have done the docking, right, all of you? So that is what all about from my side. I hope that you all of you get the insights for the same. If definitely if you have anybody, if any query, definitely you guys can ask for the same. But the more insights we are going to take in our internship program, we guys definitely will walk through towards like how you can do the molecular docking using AutoDock Vina, and step-by-step guidance will be given, followed with what kind of molecular visualizations can be done, and how the interpretations of the results can be even followed. So with that note, I will just finally conclude from my end. I just request other speaker to come ahead and just give a very good insights regarding the further steps. Thank you, sir.
Yeah. Thank you, Nilofer. So, good evening, everyone. I hope my slides are visible. Can anybody confirm?
Yes, sir, it is visible.
Yeah. Okay. So hello, my name is Dr. Abhishek Aher, and I'm here to present like about the case studies. As Prodyot and Dr. Nilofer has like very well presented how docking is done by like theoretically by Dr. Prodyot, and Dr. Nilofer has like a very in practically she mentioned a very easy way how we can do a basic docking without having a very high system computer using SwissDock. So coming back to the case study, so as we know, like what is HIV is like HIV is like human immunodeficiency virus. So what happens is like it's a retrovirus. So it's a retrovirus that attacks the human immune system, primarily CD4 plus T cells. It is transmitted through blood and body fluids. So what is the impact on health? So it causes immune suppression, making the body vulnerable to infections, and so that leads to further additional diseases. So what are the current treatment are like nothing but that these are ART, that is antiretroviral therapy, which is a combination of drugs which we give, and ongoing need of like innovative drugs and strategies to overcome the drug resistance. So for the bioinformatics part, for any drug, for coming to the from the maybe like the starting stage to the going back to the like clinical trial, there are mostly these 9 steps are involved. So starting from the target identification to the regulatory approval and commercialization. So these are the 5 first stage. I'll just go for the pointer. So basically, these are the 5 first stage which are mostly important, and which are the foundation for any drug to get approved, and which will not get discarded into the clinical trial stage. So I'll start with the for HIV proteases. HIV protease is nothing but aspartyl proteases, and it is one of the target for HIV. So for the target identification, we have chosen this HIV proteases. So somebody can say like HIV is nothing but is retroviridity. So why we are going for the HIV proteases and not going for this reverse transcriptase is one of the factor which Pradyot had mentioned in the first speaker, this target identification, like this HIV protease has a very symmetrical active site, which made this for which is make this more favorable for bioinformatic docking analysis, simulation analysis. But whereas this retrovirus, like reverse transcriptase enzyme, it active site is not very stable, it is very flexible. So because of that, it does not fall into a favorable target. So after the target identification, we have structure determination. So we already have the structure of the this proteases with extra diffraction method you can take, or you can take the structure from this NMR, or if you have some other structure, you can go from also different softwares are available to get the structure, which are predicted structures are also available. So for this HIV proteases, we have these people have done a lot of virtual screening, and in that virtual screening, they have found this one of the molecules. This is sequenavir, and they further optimized this molecule, and this optimized molecule is further went ahead with the in vitro validation, and in vitro validation, they found out that is showing good results and reducing the plaques formation of the HIV. And further, this again it gained for the preclinical testing and clinical trials and regulatory approval and commercialization, where it finally this is the important step where it's a final approval of the drug. So by doing this, the sequinavir is the first generation inhibitor developed using the structure-based drug designing, and this indinavir is also optimized through docking simulation for effective protease binding. So how bioinformatics can help in research? So one of my project, what we have done is more than 100 compounds we have screened for the cytotoxicity against cancer, and we found out that out of these 100 compounds, we found only 50 compounds were the one which were showing to be cytotoxicity. And out of these 50 compounds, we for these 50 compounds, we did different assays like MTT, LDH, FACS, live and dead assay, different assays to check its efficacy. And we found out that out of these 6 compounds, based on whether it is effective against the primary cells or not, and obviously using this pharmacokinetic and pharmacodynamic properties, we found out only this X molecule was shown to be the best molecule among them. So, but for this process, we took almost two years it took for us. But further, if we would have done with the Swiss ADME, the same 100 compounds I have taken, and I have run this Swiss, and I put all the structures into the Swiss ADME, and we were able to find out based on the drug likeliness and based on the Lipinski's rule of 5, which is a important tool for any molecule to be a drug. It should follow the Lipinski's rule, Goh's rule. Based on this molecule, we were able to find out that among these 100 molecules, the one molecule which was also coming through in vitro is same which is coming from in silico data. So as one person can tell, only instead of going from this 100 molecule following this in vitro, if we could have gone directly with the Swiss ADME, we got only this single molecule, then we could have saved 2 years. So that's a simple example I'm giving. Obviously, we had to go through it anyhow, so that's why we went for the normal in vitro method. So we can see in Swiss ADME, there are two, one of the, what it shows is this is a bioradar. So based on the lipophilicity, size and polarity of the molecule, we can see if a drug is falling into this pink allowed region, this is a more drug likeliness molecule. So one molecule which we found is, this is the one, and which is showing the best result, which was following all the drug likeliness drug likeliness properties, and which was also falling within the bioradar. Another is this is another is the best figure we can get from the Swiss ADM is like the egg plot. So why it is egg plot? You can see this is the egg white egg part, and this is the yellow, but this is a boiled egg we call it. So here you can see this yellow region is called the BBB region. That is nothing but blood brain barrier region. So any compound, let's say if you're any drug you're going for, maybe you are going for maybe liver drug, or maybe any drug which is going for, maybe you are some which is something acting on your lungs. So you don't want your drug to go to the brain. So any drug which is not actually required to go to the brain shouldn't be a blood-brain barrier crossing drug. So I was able to, in my cancer drugs, I was able to find out that, say, among all those drugs, these drugs were not going inside or crossing the blood-brain barrier region. Also, one of the thing you can see, this is the white region is nothing but human intestinal absorption region. So if a drug has to be a good drug, it should also be ingested through orally, and that ingested drug should also should be absorbed by our human intestine. So you can see these drugs are the one which can be taken orally, and these drugs can be absorbed through human intestinal gastrointestinal absorption. So one of the another thing, like one person can also say like, okay, so they we know that this AB like, okay, your drug is getting absorbed into the bloodstream, and it maybe it is going to the cell, but there are some transporters are there. These are the P glycoprotein transporter. This is so P glycoprotein is a somewhat fancy name, based a well-known or famous name for this P glycoprotein is like ABC transporters. So what these ABC transporters are do, like any, so drug resistance is happening. We are taking some drugs and some drug resistance is happening. How these drugs, how these cells are getting resistant to drug is because of these ABC transporters. So if your molecule is interacting with the ABC transporters, or we can say if your molecule is a substrate of this ABC, that molecule will give to drug resistance. So for our sake, if a molecule is a Pgp minus, that molecule will not give any drug resistance. So among these four molecules also, you can see these two molecules are the substrate of ABC transporters. But here you can see this for the red, which is showing which is not a P glycoprotein substrate. That means these molecules are not substrate for the ABC, and that will show that this will not give to drug resistance. So, what is artificial intelligence and machine learning integration in drug discovery? So, why nowadays, because of this artificial intelligence, this is a revolutionizing the drug discovery. Because of this AI and ML, making drug discovery is very much faster. Now, as Prodyot said, earlier it used to take maybe 2 years or something, but now because of this AI and ML, it is taking very less time. So one of the, like, because of this AI and ML, best thing you people have heard within the year or two, that is the AlphaFold. So in AlphaFold, what is happening? What AlphaFold do is like it uses machine learning, and based on the machine learning, based on the primary structure, so primary like amino acid sequence of any protein, it makes its three-dimensional structure. So that is what AlphaFold do. So so like that is what do so break so that is the breakthrough, like so any till 2 years back, one of the bottleneck in our discovery was like there was no structures of available proteins. A lot of structures were not available because of their only primary amino acid sequence was available, but therefore that structure was not available. But because of the AlphaFold, all the proteins now got their new structure. So there is also, because of this deep learning and drug designing is also available. Why this deep learning is used? Because binding affinity prediction is now even more improved, and this deep learning is also used for this virtual screening. So as I mentioned earlier, like now it is because of this AI, the virtual screening part is reduced, its timing, and now it is even faster than the before. And de novo drug designing is also available. So this AI model analyzes complex biological data, recognizes pattern, and predicts drug targets interaction, and improve accuracy in lead selection, and reduces failure rates. So what AI do is like it's AI based AI is helped by machine learning. So what happened? We give a data to the AI, and machine learning what do, it recognizes the pattern. So once first maybe AI, the machine learning will be slower, but as you're giving it to data, more and more data, its accuracy will increase. So basically what will happen for teaching any AI, what we will do, we will give you a sample. Let's say that we'll go with the lysozyme in water, or maybe we'll go for this protease, and we'll this HIV protease we will give to the this machine learning, and we will say like this is the darunavir, or maybe indinavir. These are the two antiviral drugs which are showing good results against this HIV proteases. Similarly, so like based on that molecule, it will analyze all the parameter, and based on this algorithm, it will find out how to increase the efficiency and speed of this machine learning. So yeah, so what are the limitation? As I mentioned, limitation, we can see this is the you can see. So one of the integration limitation is like integration with AI and ML is that biasness. If there is any gap in the data sets, you can see a normal robot will be go wrong. So based on this, so for the AI, it is very important to have this biasness should be observed. Also, we can see, as we mentioned, for this molecular docking and everything, this rigid receptor assumption is there. As we know, in biological, there is induced fit model is also there. Or sometimes what happen, a binding of your ligand molecule to a protein, what will happen, there will be conformational changes happening, and because of this conformational change, there might be a so, but in our bioinformatics only, basically in molecular dynamic, we consider that the protein is very rigid, but in biological the protein is not rigid. Also, in molecular docking, what we do is like we do docking without involving the water molecules. So there is inadequate solvation is there, and entropic consideration. So what happen, normally, basically in molecular dynamics, not molecular docking only, what happen, we are only taking a protein and we are removing all the waters and we are taking the ligand molecule. But in our in vivo system, what is happening, there is also water molecules are surrounded to the protein. Also, there are different ions are involved. Because of that, the physiological parameters are changes for protein to protein. So, and another thing, like for any computational, for virtual screening and everything of maybe structure-based drug designing or ligand-based drug designing also, the quality of the structure data is the one of the important parameter. If there is a issue, maybe in the X-ray diffraction or maybe NMR, if there some of the scientists have made some mistake, or they were not able to find a proper structure. Because of that, the quality of the structure is disturbed, and based on that, so this AI will not be able to find out whether there, or molecular dynamic will not be able to find out whether there will be any how to say, there will be any false positive results are there. Based on the foundation you have been set, it will go for that ahead. So quality of the structured data plays a very important role. And again, this is polypharmacology challenges. So what we are considering is like, let's say this darunavir or maybe indivir or sequinavir, we are taking this antiviral drug, and we are directly targeting it to this protease of HIV. But we are not expecting that there are other proteins, other pharmacophore, which are also present inside the human cell, and these antiviral drugs might or may interact with these molecules also, and which may give us false positive result. Or sometimes what is happening, these molecules are also interacting with different molecule, which are like two molecules cumulatively giving effect to this protease inhibition. So that is also available. So these are the limitations for the molecular dynamics and molecular docking. Basically, dynamics is somewhat different. So what are the future direction in drug discovery? So one of the thing is like for future direction in AI is like now it's coming nowadays is like quantum computing. So how quantum computing is different now? The current available of this computer is like, now current available is like binary. So now how it is happening, like there is only one or zero. Is it yes or no? These are the two options are available for the current computers. But what is happening in the quantum computing is like, instead of one or two, but in quantum computing it is like one and two. It is happening simultaneously. So a computer, now there is a new terminology, superposition is happening. Now computer can think there's one and two are happening simultaneously. So because of this thinking, so there now computer has a now it can solve, because of this quantum computing, it can solve different complex problems also. So now this is the new future which is happening in the drug discovery. So mostly you can see the industries are the one which are like starting developing this quantum computing in the AI based drug discovery. So advancement in the multi-target modeling and AI-driven toxicity prediction and systemic biology approaches will be enhance the understanding of drug actions in organism level, paving the way for safer and more effective and personalized therapeutics. So as I mentioned earlier, so we now the AI like as also incorporating so all the pipeline. So basically, one of the so normally if you are going for a AI for virtual screening, first of the time what will we do, we'll go for the directly virtual screening, maybe what are the best molecule we are getting, those molecules will go for the ADMET Swiss ADME studies. But now because of this AI, it will go for extensively checking the drug likeliness and the ADMET properties of the molecule, or whether it will also check whether it's called molecule is crossing the blood-brain barrier, or whether this molecule is also already present for any repurposing. So based on this data, these newer models of this AI ML will increase its drug discovery even further than it is available. So I think that much from my side. Thank you.
Thank you so much, Dr. Abhishek. So that was a very informative session. So we are running out of time actually, so we will not be able to have a Q&A session, but if you have any questions, please leave it at info@biotechnika.org and we will get back to you. So I think it was a very nice session. We got to see firsthand how these tools work, especially what Dr. Nilofer had shown us. So it was the first time even I'm looking at something like that. So it was a very nice experience. So a big thank you to our speakers for sharing their expertise, and also for our audience for joining us today. And special thanks to Dr. Abhishek for taking the time out of his busy schedule to do this for us today. And guys, if you want to know more about this molecular docking and stuff, like how Dr. Nilofer ma'am had already shared, you can check out the BGTI training, that is Global Tools and Techniques of Bioinformatics, which is going to start from next week, that is next Tuesday. The link is in the comment section, so do enroll and you'll get to know more about this, and you'll see hands-on how it is, and she will guide you and you'll have a walkthrough of it. So thank you so much for joining us, and we hope this session has provided valuable insights into the bioinformatics field that is driven by drug discovery. So thanks you once again, and we'll see you in our next session. Good night and take care, everybody.